very much. Thank you for staying to the final talk, and thank you very much to the organizers both for inviting me and for arranging for there to be drinks after the final talk so that people would stay to hear it. And I've enjoyed very much being at this conference, and sometimes, you know, depending on which audience I'm talking to, I have to explain why robots, why bio-robots, etc. But I think you guys all get that part. What's probably the main uppermost issue in the head of all of you is why insects. So I just want to give you a few reasons why insects. So many of us here work on bio-inspired robots, often humanoid robots, but also, you know, we know quite a lot of other quite impressive bio-inspired robots, quadrupeds, swimming robots. But when you look at living things on this planet, the vertebrates make up a fraction of a % of the living things on this planet. Plants and algae, microorganisms, and then invertebrates are pretty much the success story of life on this planet. And that's you know, if we do it by the number of species that there are. If we do it by the proportion of biomass, they win even better. So they're doing something right. And maybe we can learn something for robotics from insects. The other thing that motivates me quite a lot is the human brain is just too complex. And the insect brain, by comparison, is very small and relatively simple. So from the talk we had from DeepMind, it's very clear that we have the computational power to model the brain of an insect with maybe 1 million neurons and probably fewer than that in many insects. So we should be able to do it. And the reason we can't do it is not because we don't have the computational power, it's because we don't understand how the brain works. Well, it feels like something we might have a hope of understanding, unlike, at least from my point of view, unlike the human brain, which is really complex. So an insect brain, you know, literally fits on the head of a pin. The other analogy I often use for the size of the insect brain is that an insect brain would fit within 1 voxel of an fMRI scan. So if you put an insect in the fMRI scanner, you'd know if its brain was on or off. That's the only information. All right. So the other thing the other motivation, obviously, is that insects do a lot of interesting things. And very fortunately for me, Lars' talk this morning hopefully convinced you that insects do a lot of interesting things. So I'm just going to focus on one particular thing, which is also quite useful as a kind of introduction to the type of methodology that I use as well in the work that I do. So this is a cricket in a little experimental situation where the ground here is heated up. So there's one cool spot, but you can't see it because we don't want there to be any cue for the cricket either about where the cool spot is. It can only find it by searching around. And many of you might know the Morris water maze, which people have used for many years to look at cognition in rats, spatial cognition in rats, where you put a rat in a vat of water and it can't see the platform under the surface and it has to swim around. And you often have these, and you have cues on the side. So you can see there, it finally found it. By the time you get to trial 6, it's looking like it's doing something quite different to what it was doing on trial 1, where it was, you know, trying to escape and so on. And in fact, it's actually going quite quickly to the cool spot. And then I think we have maybe one more, or that might be my short version of this video. And once it finds the cool spot, it stays there because it's definitely much more pleasant. Okay, so we can obviously run this with multiple crickets and see what they do. This experiment was first done by Mitsunami with cockroaches, and then we replicated it with crickets, and then more recently has been done with Drosophila, and they do the same. Oops, sorry, that's not what I wanted to do. There we are. So if we do it with lots of crickets, we see that the time it takes them to find the cool spot decreases, although it is quite noisy. There's a few who just never get it. Interestingly, if this is with these very artificial cues, if we use cues that are more like natural scenery, then they do much better. They actually learn it faster, and it's much a higher proportion learn it, and they get much quicker. So it seems like something that they really can learn. In the rat, this is a classic experiment where if you damage the hippocampus, they can't do it. insects don't have a hippocampus, but they can still do this behavior. Okay. And the kind of approach we take to try and understand this is to say, well, let's try and make a robot do the same thing. So this is the robot in this case. It's a small, you might recognize it as a Kepler robot underneath. It actually has polarization sensors, which we're not going to use here, but it has an upward pointing camera that looks at a mirror. It's not a very expensive one. It was actually a doorknob. But it gave us a nice 360-degree view, which is what insects have, more or less. And then we could basically put this into a framework for comparing different visual homing models. So basically, we get visual input. We could put this through a little cricket eye model to make it quite blurry, because crickets have relatively low resolution vision. We could test it in some algorithm to say, how do you try and get back to the cool spot? And just to cut this part fairly short, we actually had this target behavior, which is, if we make them do this in the dark, they're really bad at it, not surprisingly. If they have distinct landmarks, they do quite well. If they have a natural scene, I just told you earlier, they do better. Confusingly, we initially had blank walls as our control, and the crickets were learning it with the blank walls, which was a bit of a worry. But it turns out they weren't as blank as we thought they were. There was a slight light gradient, and that turned out to be enough. And the actual algorithm that worked is to use a binary difference algorithm. So basically, when you find a safe spot the first time, you take a snapshot, effectively, of that place. You say, what does it look like? And you store that image. And then anywhere else in the arena, you take an image, you convert it to black and white, so it's simple pixel, 0-1 comparison, and you just take the difference of the picture that you saw when you stored it versus what you see otherwise. And if you're actually in exactly the same spot, then you'll get a 0, so that's what this sort of surface shows. And as you move away from that exact spot, you get this actual smooth surface going away. And then you can just use a very simple gradient descent of any kind you like for the motor output. You just run down the slope, basically, and that will get you there. And it turns out that indeed the natural scene had a very good surface. It's a little bit less good in the artificial landmarks. And then the black and white image, the blank wall scene turned out to actually have a fairly good information as well. There was actually a basin that the system could find, so we could replicate that. So that just kind of gives you the flavor of how we try to go about this work, is to try to understand what the algorithm is by implementing it on a robot. And several of the ideas that I mentioned here are going to come back in what I'm going to talk about. So the main additional thing that sort of happened in more recent work that we've been doing is we've tried to get these algorithms to really be closely based on the insect brain. And when I say closely based, we're trying to build models which have one-to-one correspondence. This is an identified neuron in the animal. We have that neuron in our model. That's the kind of correspondence we're going for. Okay. But let's motivate this a little bit more by what insects are good at. So this is just the same problem, but now in a very, really everything, a really natural scene. So it's not just an arena with a kind of natural scene, but the animal is flying freely around and it has to find its nest. So these are wasp nests. And you can see that it's pretty hard to locate, but wasps are very, very good at being able to find them again. So they can do very long journeys away and they come back using path integration, but that always accumulates error. So for the last few meters, they really have to depend on visual memory to do it. But they're in these very highly aliased surroundings. So that they're able to pinpoint the entrance. And then another insect that I've looked at a lot is desert ants. So this is a beautiful video taken by Antoine Wistrak, who was a postdoc with me. It's a desert ant running home, and hopefully one thing you can notice here, it's not doing chemical trail following, it's not antennating the ground or anything like that. Each of these ants operates individually, and they have good navigation, and the navigation is crucial to the colony's survival, and they've been observed to do this distances as far away as 1 kilometer from their nest and get back home again. And as in this example, they can do this both in very flat and empty environments, but also in these very complex, cluttered terrain. And the two mechanisms which have been known for a long time that they use are path integration, odometry, dead reckoning, and visual memory. And I'm going to be focusing on the second one of those today, but there's a very, very interesting story about the path integration as well, so I'm very happy to talk about that afterwards if people are interested. I should probably say, I meant to say at the beginning, I'm actually happy to take questions as we go along, as well as at the end. So if at any point people are feeling like, I don't understand, please put your hand up and I'm happy to take questions. I'm sure we can balance that out in the timing, so that's good. Okay, so we do field studies of ant behavior in a nice abandoned industrial site near Sevilla in Spain, which is good because these desert ants operate in the hottest part of the day, in the hottest part of summer, when they can scavenge on everything else that has died out there. And apart from, and that's not the time of day when any Spaniard would be out, but it's when we're all out doing our experiments. And then we can go back to Sevilla and have tapas in the evening, so it's not so bad. So what we did in this set of experiments, we were actually following ants around with a differential GPS and a downward pointing camera. So this is Mike Mangan, another former postdoc, PhD and postdoc with me, doing that. And this is the area, so we have control over when the ants come in and out of the nest. We put a little kind of plastic shield around the nest so that we can let them in and out, and we can mark individual ants so we know when individual ants are coming in and out. So it looks a bit like this. So that's Mike trying to follow the ant, and occasionally someone pointing out, no, the ant's over here. And we tried to follow these ants from the first time they ever came out of the nest and to see exactly what these ants experienced. So a little bit like Lars was talking about for the bees earlier. And then Benjamin Risser, another great postdoc that I had, did this amazing tracking algorithm. So this is a moving camera, unknown movement of the camera. The ant is this tiny little spot there, occupying a few pixels, and it's this very complicated terrain. And he managed to come up with this fantastic tracking algorithm that even when the ant gets lost, it can disappears under the leaves. It can make a really good estimate of where the ant probably is. So that was nice. And then the other thing we also did with this was obviously we try and reconstruct what the ant's eye point of view is. So given where the ant is, what does it see? And here you can see, again, we have this kind of lower resolution of the ant eye. And we also, well, you could just see briefly there, you can maybe see it down here. We have this pattern in the sky, which is this polarization pattern, which ants are sensitive to. So we're trying to kind of imitate that as well, their kind of color space and their ability to detect polarized light. Okay, and just to show you again, so this was this whole experiment. We tracked 14 ants, and then with Lars Hauk and Benjamin, we did this fantastic reconstruction of everywhere the ants went. So basically reconstructed the whole ground surface. So this is an 8-meter circle where the ants were traveling. Okay. And then, but we can zoom in and see really closely. We can see things like, are the ants going around bushes, or how much does the same path of the same ant overlap on each excursion it's making. You can zoom in even more and look at the actual terrain. If it's slowed down a bit, is it because it's slowed down because it's not sure where it is, or has it slowed down just because it's got caught in some kind of rough terrain? So this has been really fun data set to work with. And this is just one example of one ant. So this is a naive ant. It's the first time it's ever come out of the nest, and it makes a foraging excursion, shown in blue here, out to point 1, and then it gets some food when it's about 8 meters away from the nest, and it runs straight home, and hopefully you can see that it doesn't go home on the route it went out. It goes pretty much straight, and we know that it's using path integration to do this. We didn't in this experiment, but people have done a lot of experiments where they pick up the ant at this point and move it somewhere else, and it will run straight down and then start searching, so it can go straight back to the nest. They also, when they get back to the nest, they know where that food was, because when they come out again, the second time, they go straight back to where that food was. Okay. And then they come home again. And what I want you to notice here is when they come home again, they're following a very, very similar route to get back home. So what we did then was to interfere with this poor ant and to, oh sorry, yeah, I'm just going to mention, the path integration, which I am not going to talk about anymore, is this paper is the kind of the key paper where we talk about the neural mechanisms of that. So, but what we did with the ant was then after it had just been twice, so it had gone out, come back, gone out, come back, on the second time, before it even got into the nest, we would pick it up and put it back to this food position and look at what it did. And what it does is to do a little search, but then it basically finds the path and it follows again pretty much the same path. So here it diverges from the path it took before, and then it finds it again and follows it very well. It diverges a little, finds it again. And here, because we have this really detailed tracking information, we can actually say, you know, what's the forward velocity, and for example, we can see that when it comes off, it speeds up when it's on the path, and then when it comes off, it starts to slow down. The bigger blobs means it's also got a higher angular velocity. And you can really see here, for example, that it comes off, it slows down, it starts to look around, and then suddenly it finds the path again and it follows it in. So, and again, this is something, you know, that's being known, but we've not been able to show in such detail that they really follow very much the same path. And this path will be different for individual ants. So each ant will find its own path back. It has to dodge some bushes and things. A different ant will dodge the bushes a different way, but then they'll be very consistent on these paths. And from the ant's point of view, this seems like a difficult task. So this is, again, pictures from kind of on the ground and a reconstruction of what it looks like, and then what it looks like with ant eye resolution. So basically, the task you're given is you watch this video twice, and then you have to use this to get home again the third time. Okay.
I'm lost.
Yeah, it's pretty difficult, I think. So that's the problem that the ant seems to be able to solve. So the question is, you know, what memory does it need to do that? So this is not something that can be innate to the ant. The ant has to learn something because it doesn't know what its environment is going to contain, but it needs to learn it very quickly. And a nice suggestion that was made by Bart Badley et al. In 2012 is that maybe to follow this route, the ant doesn't have to recognize every single location along the route, but what it needs to do is just recognize that this is familiar. So it doesn't need to know, okay, I was 3/4 of the way along the route when I saw this. It just needs to know this is something I saw before. And the analogy Bart often uses is, you know, that you're in a foreign city and you're wandering around and you're getting a bit lost, but then suddenly you turn the corner and you see something that's familiar and you think, ah, good, I am going the right way because this is familiar. So the algorithm that he proposed was that basically the first time you go along the route, which is the first time you're getting home, it's being guided by path integration, you should just store everything you see. So for example, just regularly every 10 centimeters on the route, store that image of the view. And then when you're trying to go back again, maybe without your path integrator because a nasty experimenter has picked you up and moved you, so you don't know, you can't use your path integration, then at every point you can essentially stop and scan left and right and say, when does my view match? So I'm just doing a simple, again, pixel comparison of what I see to what I saw before. And that's what's kind of illustrated here. So this is 8 meters along the route. This is the direction you were facing when you stored that image, and this is what you get from this pixel comparison to the image difference as your heading angle changes from the heading angle when you stored the route. So you might have done a complicated route that we kind of adjusted the 0 to be always the same. So what you can see is that you basically get this valley that you can follow to go along the route in a very similar way as we did the gradient descent before. So now it's the same principle, but now extended with a large number of images. Okay, and quite importantly here, there's no sequence to these images. You just store all of them, and when you're making this comparison, you just compare what you see now to everything that you stored. Okay, and it turns out this works quite well in simulation and on robots. Oops. But obviously the question is, well, how could the tiny brain of an ant implement an algorithm like this? Okay. So what what we're asking the ant to do is to associate the views that it experiences with its progress towards the nest, and it also builds up one of these routes going out as well. So it builds up a distinct route for going towards the food and one for going towards the nest. So it needs to make this kind of association. And that obviously hints that what it's doing is some kind of visual memory that involves pattern recognition, and we have a lot of information actually about how learning of sensory patterns might happen in insect brains. And there's a particular area of the insect brain that's specialized for olfactory pattern learning, which is the mushroom bodies, and this is, some of you may have come across this before, that's been studied quite a lot. But in ants, this area also has extensive visual input. So just to give you, I'm assuming not everybody's an expert on insect brain anatomy, so to just give you a little bit of guidance, this is a nice picture from Heisenberg 2003. So these are the eyes, this is a fly, the eyes. These parts here are the visual processing areas, which are quite large. And then there's these two structures here and here, which are called the mushroom bodies because they look a bit like mushrooms. It depends on the species how much they look like mushrooms. They look even more like mushrooms in flies and ants. In bees and ants. And then there's a number of others. So this is the olfactory center. There's this area in the middle called the central complex, which is very interesting, but I'm not going to talk about today. So as I sort of said, in ants, so for example in Cataglyphus nodus, which is a close relative of the species we study, there's a lot of visual input into these mushroom bodies. So our basic approach here is being to say, let's build computational models of this brain area and then test to see whether that neural circuit can actually support these navigational tasks. So again, as I said, what we know most about in this system is actually olfactory learning. And again, this is a figure from Heisenberg, but it's, adapted from Heisenberg, but it's a good way to kind of explain the basic principle here. So you have input from odors to the antennal lobe, and they get processed by projection neurons, which send axons up to the mushroom body. So this part is the mushroom body, which consists of lots and lots of Kenyan cells. So these are many, many cells with lots of parallel fibers. And I've drawn them all as all going just together. They actually go together and then they split into several lobes as well. But for the moment, we can just assume there's just lots and lots of parallel fibers. And the key thing here is that you have some kind of pattern here, based on the odor input that gets projected into a sparse pattern in the Kenyan cells. So that for any particular odor or odor combination or odor experience, you're going to get a quite specific small set of Kenyan cells that are active. And the main learning mechanism that's known, for example, if you pair that odor with a nasty shock, is that the synapses between those Kenyan cells and a particular mushroom output neuron, mushroom body output neuron, of which there's much fewer, like around 20 to 30, compared to the hundreds of thousands of Kenyan cells in the bee and tens of thousands in the fly. So basically, each synapse of the Kenyan cell gets changed in weight. And in fact, what happens is that it gets turned down in weight. So you could imagine, an easy way to think of it is these are all initially active synapses, and then when you want to learn that this odor is bad, what you do is decrease the weight in those synapses of the Kenyan cells that are on. Okay? So that means if you ever see that same thing again, experience the same thing again, the output here will be lower. So you'll know that it was a bad thing. And for example, if this could be something that drives you towards odors and you stop going towards the odors because it's a bad thing. And then you can have the vice versa, you can have another output neuron which is coding the good things, and you turn and act, but actually drives avoidance behavior, so you want to stop doing avoidance behavior to things that are good, so again you turn them down. And I've just written here, because it will be useful later, this is essentially the learning rule. Okay, so we just change the weight according to what happened. These neurons here are dopamine neurons, so you have the reward is activating dopamine neurons, And they're very distinctly paired in this mushroom body. Each dopamine neuron is paired with a particular mushroom body output neuron. So when you get the shock and the odor together, then those two things cause a decrease in the weight. Okay, so it's Kenyan cell activity and dopamine release makes it go down. And I just want to point out here that this is not Hebbian learning. Okay, it's not STTP. It doesn't matter whether this mushroom body neuron fires or doesn't fire, and nor is it prediction error learning as is used in typical RL. But there's overwhelming evidence that this is what, at least one of the mechanisms that's causing change in the synapses in the mushroom body of insects for olfactory learning. So let's go back to our visual learning. So we basically had already built a model of this olfactory learning, and we decided, well, maybe we could just use this for visual learning in a really, essentially very stupid way, is to just say, well, what if the visual input also is just a pattern? So let's just stick that in there and have a set of Kenyan cells that respond to the visual stuff, a set that still have a set that respond to olfactory stuff. There could even be some bimodal responsive cells. And here we didn't even do any fancy visual processing. We just literally took the intensity values of each of the pixels as our pattern of input in the projection neurons. And then we did, again, a sparse projection into the visual neurons. And we did exactly the same thing that we now we're just learning, if I'm going home, then I should learn those images. I'll change those, the weights for those active cells. And if I'm going out towards the food, then I'll learn this set. So that then basically the output of the neuron it's actually the opposite of familiarity. So if the neuron is spiking, it means you haven't seen it before because we're turning things down. So basically, as long as the neuron doesn't spike, it means this is something where the weights have been lowered, which means it's something you've stored already. Okay? Does that make sense? Yep.
The representation should be rotation invariant, right? Because the ant is coming back, the pattern is different, even though it's in the same place.
No. So two things there. So we're actually assuming here that they're learning a completely different set so they have a different output neuron for the two things. So they're learning one set of views for going home and a completely separate set of views for going out. And actually there is evidence that they
cannot recognize they have seen that place.
So exactly. So if and there's actually evidence that if they're carrying food and you put them on their outward route, they don't seem to know where they are. And then if you take the food away from them so that they're foraging again, they recognize where it is. Yeah. And the other thing is that it's also not rotation invariant, although we'll come back to this point, because part of the mechanism that we're using to actually use this is to say, I want to align myself by rotating. When I rotate, when I get the dip in the output, that's the direction that I want to go. Yeah. So you're actually using the rotation is part of the information you're using. Yeah.
Sorry, can I?
Yeah, sure.
The part on the polarization of it's not captured here.
No.
It's a different.
Yes and no. So generally, no, as in most of the time when we're processing these views, we haven't been including polarization in there. Polarization is used in the whole path integration system, and that's, as I said, another story that I could give you a whole talk about that, but I'm not going to. So here we tried both ways. Sometimes we included it in the view, and sometimes we didn't. So, but yeah, it's optional. Okay, and this actually works quite well. Again, so this is a little simulation of. There we go. Of what the ant is seeing. So this is what we're learning. Is about 100 of these views along the route, and then we're basically putting the ant back and saying, can you follow the way home? Where either you just try and go roughly in the right direction within plus or minus 60 degrees, which obviously doesn't do very well, you make a lot of errors, or you have this, what I call perfect memory, which is where you've literally stored the images and you're doing image comparison. So you have to sort through, you have to have all the images individually stored and sort through them, or you have this mushroom body model where basically each image is stored on top of another one, but in the end, the output you get is actually very good, and you can follow very well. And an obvious question people have here is, well, what's the memory capacity? Aren't you just going to run out of memory if you're just turning weights down, and you're not, at the moment, in this model anyway, we're not adapting anything. How many things can you do? So one thing that's quite interesting, as I said, because it's a sparse code, it means most Kenyan cells will actually be active only when you're in one specific place. It turns out that if you track one Kenyan cell, it's very rarely active, which means that if it is active again, you're probably in the same place, facing the same way. And we can make this whole thing a little bit more abstract, where we literally just have one-shot episodic learning of the value, and we just have everything be zeros and ones. And if you do that, you actually end up with something that's basically identical to a Wilshire net, if any of you remember Wilshire nets from back in the day, which are a type of auto-associative network. But if you simplify it in this kind of binary abstraction, you can actually get an analytical solution for the memory capacity, which is kind of shown here. So basically we calculate how many random patterns could you store before the probability of a new pattern being incorrectly recognized is above 0.01. And it turns out that's quite a large number. I'm not sure if you can see the numbers here. There's 1,000 there. So it depends on how many neurons you have in the KC layer, and it depends on the sparsity of encoding. Certainly for the number of neurons that we know that ants and bees have, and for reasonable ideas of sparsity, you're certainly up to being able to store hundreds of different images before you hit this barrier. And that's not making any effort to do anything efficient or clever about the encoding. And there's actually been a lot of work on sparse encoding and on so forth, where you can try and make that more efficient as well. So we think it's actually quite plausible as a model for the ant. It's sufficient for what it has to do. And we've actually, of course, tried to put this on a robot. So this was work by an undergraduate student, actually, who made this little robot, which is literally a mobile phone on wheels, which was nice because we just stuck a omnidirectional lens on the camera for the mobile phone, and then we could communicate with it very easily. He just built a little system to be able to have it control the wheels. It has its own Gmail account. So basically we have this low sampled image, and then we're trying to do this comparison and decide which way to go. And here the algorithm he was using for this particular part was saying basically oscillate proportionally to how familiar things are. So if things are familiar, then just go straight, and if they get less familiar, then oscillate more and slow down. And then we also took it out to the desert and tested it also in the desert. So the main thing we had to put flat boards because it couldn't go over the rough ground that the insects were on, but otherwise it was able to follow routes in this real situation. And to go back also to our wasps, we also tried to do this on a aerial vehicle where now we're trying to again pinpoint a single location. And the idea is that this is your home, and as you fly out, you store, again, images as you fly out. And when you come back, we use this strategy of just trying to cross over. So you try and keep, and if you imagine a flying insect, it can keep heading in the right direction. And now when it's saying, when I cross over, does this match anything that I saw before? So this is what it looks like in practice. So this is basically comparing all the inbound images to all the outbound images and looking for similarity. And what you can see is there's these peaks of similarity which correspond to the points at which you're crossing. So you can actually, again, know, for example, you can slow down how fast you're zigzagging the closer you get to the goal. Or, well, so I think the algorithm here was to say, if you cross, then you should turn and come back again, and if you cross, then you should turn and come back again, and so on. So that also worked quite well, and so this is real experiments with the flying vehicle, where we basically had this algorithm that tried to kind of slow down as it got closer, because in fact, as you can see, although I talked earlier, I said, we don't necessarily use the information, you don't have to use the information about where you saw something. you do actually have that information, because when you get a match, the best match is actually in the same location. So you can know that if you did store these in sequence, you would know which one is closer to the nest or not. So this is just some results from field experiments, and then we did lots of realistic simulations, including one of it with a simulated world where we do these ant experiments and several other simulated worlds. And here, just to emphasize, here the camera is looking down. So what we're learning is what the view of the ground looks like, instead of what the surrounding view looks like. So in both these cases, you can use this method quite effectively. Okay. So I'm doing okay for time. So what I've told you so far about the mushroom body is simplified. And so what we've been working on recently is taking into account some of the more complex information that we're actually finding out about the mushroom body. So there's been a lot of neuroanatomical and neuroimaging methods that show that the whole structure is a bit more complex. There's actually now a complete connectome in Drosophila for this structure, where by connectome we actually know every single synapse, how many synapses there are between any two neurons in the system. And then using neurogenetic methods, you can actually selectively activate or silence or record from individual neurons. So you can just basically say, well, I want this particular output neuron. I just want it to light up when it's active, and I'm doing that here with a animal that's on a little trackball. It's behaving, and you can see that neuron's activity. And then you can do another genetic manipulation to look at another neuron's activity, and so on. So it's really just amazing stuff that we can use in our models now. So I'm just going to cover a couple of different things that we've started to work on, and I thought I should include this one, given that this conference includes development. So one thing, so I mentioned that the connectivity in the mushroom body involves projecting these sensory input patterns into a high-dimensional sparse coding. And there's actually increasing evidence that this is an adaptive process, and in particular it's a developmental process. So that what happens during development is that each of these Kenyan cells turns up a few at a time. So there's just a few neuroblasts, and gradually the animal develops more and more and more of these Kenyan cells, and they wire themselves up to the projection neurons as they arrive, basically. So we've actually recently been exploring a few models based on this, where we just introduce developmental wiring. So basically, we assume the animal is getting input, so it's learning about its world in some way. And each time it gets an input, and each time it develops a new Kenyan cell, it just says, well, what's the most active input at the moment? I'll wire to the top N PN neurons, projection neurons, at that point in time. So we can either just do that very simple thing, or we can say, I will wire to whatever's most active unless there's already a Kenyan cell that's responding, which means I don't need to represent that pattern because the pattern's already there. So basically it means you're just adapting this wiring so that you're matching patterns that really occur in the world, rather than using random connections, for example, where there might be a whole space of your encoding that you never ever use because the world is never like that. So, the sky is never below the ground or something like that. So so this makes it much more efficient in the encoding. And we've actually tested this as well in just kind of standard computer vision image data sets to do retrieval of similar images, and it turns out it works quite nicely for that as well. And I just say, this morning I found out it had been accepted for Europe. So if you want to read more of it, get in touch. As I also mentioned, there's a lot more structure. Okay. In particular, so I just sort of showed two mushroom body output neurons. There's actually, as I said, 20 to 30, depending on the animal. I think up to 100 in the bee. And interestingly, so this is what this horrible thing illustrates, and you don't need to understand it. The key thing to understand is that there's a lot of recurrent connections. So what happens a lot is that these mushroom body output neurons connect back to these dopaminergic neurons, or sometimes they actually connect back to the KC inputs, or sometimes they connect to each other. So there's actually a lot of different complex connections going on there. So I think that's all I'll say rather than try and explain anything in this particular diagram. So we're gradually, rather than try and just throw all these connections in there and try and make sense of it, we're trying to gradually piece together little sub-circuits of this and see if we can understand what they might be doing. So this is one model that we published recently that tries to look at this problem and basically looks at the fact that there's these kind of feedback connections. So this is an output neuron, it's feeding back to inhibit this dopamine neuron. And you might think this should be like a perfect little prediction error circuit, because as this one learns, it inhibits the reward so that you get the difference. But in fact, as I said, the neuron actually, the activation of the dopamine actually decreases the weight of the Kenyan cell onto here, and that means that you actually have a positive feedback loop, as in the more you've got having a negative change here that's causing an inhibition here. So actually it's not a prediction error again. And then again, in the interest of time, I'm not going to try and explain all of this, but there's some really interesting interactions. So for example, that some mushroom body output neurons are inhibiting other ones. So this one that increases attraction, then if you increase attraction, you automatically try and inhibit avoidance as well. But then that kicks off activation of other dopamine neurons, in particular ones that seem to feed into neurons that we think are involved in long-term memory. So what this whole system does is to actually transfer, you learn things very quickly, but in a weak way, and then over time the whole system itself will transfer that into a long-term memory. And even wipe out, you have these forgetting neurons that will wipe out the original memory as well over time. So, and I really more want to kind of give you the flavor of this, where these, as I said, each of these neurons, we know that neuron. We can say this is the neuron in the insect that we think is having this role in the circuit. And again, I think in the interest of time, I might skip that, but we basically have a very slightly modified learning rule here, which involves looking at the current value of the weight compared to a kind of resting level of the weight. But that actually allows us to get a lot of very interesting different effects in this system when coupled with this feedback from the embryons to the DANs. And we can do experiments, with two odors getting punishment and either one of them being punished or getting a positive reward. And we can look at what happens, if we just have the very simple forms of learning, or if we actually have the whole combined system with the short term and the long term. And just to point out one thing that happens here, for example, that here you can see one of the odors is being shocked. But in fact, what the animal learns is to avoid both the odors. It just stays on the boundary between them. Whereas with the long-term memory, it actually eventually learns that it should actually go towards the one that's not being shocked. Okay. And then another property that's being discovered in these cells is that the Kenyan cells themselves have connections between them. So just drawing like this. Which this is, the beautiful connectome information they can get. These are showing synapses between the Kenyan cells. So we've actually put this into a model to actually increase, to actually add a little bit of time into the system. So instead of just having the instantaneous, snapshot that you're learning, instead what we're learning is we're using a DVS camera input, so a dynamic vision sensor camera, which basically is a camera that tells you which pixels have changed instead of what the pixels actually are, which I think is going to be shown in this video, so let me run that. So this is the robot, and then this is, at the bottom is what the camera tells you. So basically it tells you whether there's a movement to the right or the left at any moment. So that's our input, and then we're trying to learn very short sequences of that input. So we're using a spiking neural network, and we're basically looking at when one Kenyan cell spikes just before another Kenyan cell spikes. That's a short sequence that it should learn. And we can actually learn quite well to have similar, as before we had kind of familiar moments, now we have familiar short sequences. And this can potentially be more robust over these kinds of very self-aliased kind of scenes that we're trying to move through. Okay. And one reason why we're interested in some of these things is that I've kind of been a bit casual about saying, well, the ant, is running along the ground and it's looking to the left or the right, and we can, look at how it rotates. But in fact, if you look up close at what an ant does when it's running along over slightly uneven ground with a big object, its head is all over the place. So it's not just doing this simple image comparison, because every time it goes through that, it's going to see something quite different. And moreover, ants quite commonly go backwards. So this is actually a cricket, a dead cricket. Well, it stung the cricket to death and now it's dragging it backwards. And it's going directly towards its nest, going backwards like this. But you might notice there it looks like it maybe looked around for a moment, okay? So we kind of observed this and realized this was a bit of a problem for our model. And we actually did a more systematic test of what ants going backwards do. And this is the last thing I'm going to tell you about. So this is ants allowed to leave the nest. They had this little system of channels that meant they had to run this way, then this way, this way. They got the food, then they had to run through here to get home again. And we had kind of distinctive landmarks and things around these positions. And then to do the test, what we would do is pick them up. They'd run out, so they'd learn this very well, so they had this familiar route coming back. We pick them up from here when their path integration would tell them to just run in this direction. And we put them over here where their visual information would tell them they should go in this direction, okay? And then we gave them big bits of cookie, so they had to go backwards, all right? Because if they were going forwards, they would for sure, they would always just follow again if they were carrying something forwards. But if they had a big piece of cookie, this is what happens. Oh no, I've got to show that video. So this is what happens. So it's pulling it backwards. It's going in the direction its path integration tells it, upwards. And then it has a little look around, and it goes, then changes direction, goes the other way. It was quite fun trying to find exactly the right food to make them go backward. Okay. Oops. So what we found was that basically their direction before peaking was generally the path integration direction, although quite variable. And then after peaking, they all started to go, most of them started to go in the right direction. In the peak, they line themselves up in the right direction, and then they will go in the right direction upwards. So what this suggests is that they actually, they have to somehow relate their visual recognition. So we think they still want to align with the view, so they need to align to recognize it. But once they have aligned and recognized it, they can then use that information in some much more flexible way to say, well, now when I turn back, I know which direction I want to go, possibly relative to the sky polarization. This is one of our hypotheses about it. So it's obviously, again, as with everything in nature, is more complicated than you might think. So let me go to my conclusions. So as I hope I've convinced you, insects have many skills, such as visually guided navigation, that are both cognitively interesting and go beyond the technological state of the art. And the brain side suggests this is not just that they have more computing power. And the fact that they acquire these skills very quickly suggests also that the solution is not just to throw, deep nets at it with lots of big data. So neuroscience is producing for us exquisitely detailed information about the connectivity and function of these neurons during behavior. So it's really exciting that we can now start building these one-to-one neural models and then put them on a robot and test them in real tasks, even outdoor navigation tasks. But of course, there's still many questions to answer. And so I just thank people who were in my lab, or people who are in my lab, and collaborators and funders. Thank you.